Come on. So I kept asking some people to ask me questions if they didn't know the answer. The lab is supposed to be an easy way for you to get points. If you don't know an answer, you can always look it up. And you can certainly ask me as well. Um, but people weren't looking it up and asking me, and so they weren't getting the best scores in the lab. So I'm changing the way I'm grading the lab, and it's due on the same date, but I will go through and tell you what questions are wrong and then give you, open up more time so that you can make your changes. And I think what I'll do is I'll make the changes always due on Tuesday. So the lab has been changed, and I'll talk more about that in the lab. But you can improve your score if you go in and correct your answers. Any questions about that? If not, let's go ahead and
I
get
have a question, but
started.
it's actually concerning the infectious disease project.
Can you repeat that?
I
I'm
said I
going to
actually
turn
have
up
a
the volume.
question. I have a question, but it's about the infectious disease project.
Um, Go ahead and ask.
I'm having trouble locating some information regarding the socio-political and economic concerns related to the virus. Where would be a good place to find
Hmm, I'm not real sure. What's your project on?
that? HPV?
Say again?
HPV?
Uh, well, you have to realize that the socio and political and economic information is anything to do with how people perceive HPV. Like you could go talk about religious beliefs and why some people will refuse their family members, and I guess I'm really speaking about their children getting the vaccine for that. Um, you could talk about the economics, meaning anything related to money. Where you can get maybe a point on this, let me go ahead and take a look, is you can get, oops, I can't do that. You can get the initial information at Wikipedia. The thing is that you have to back up everything you find at Wikipedia. Oh, I still don't have it here. So let's go HPV. And see if there's even a topic about that. Well, they have epidemiology. Let's see what they have about management. Yeah, all right. No, I was hoping they had a little section on it there, but I'm not finding it even on Wikipedia. History will have a little bit. Nope, it doesn't have much. All right. Let's talk about this later because this is something really for just you and me to talk about. Okay?
That's fine. Okay,
And
yeah,
I'll
that's fine. Thank
try
you.
and find something. All right, let's go ahead and start recording. Oh, actually, I'm already starting recording. Share the screen. All right, are there any other questions? If not, let's go ahead and get started. So we're we're supposed to be doing Chapter 14, Principles of Disease. We'll probably finish that lesson today, and I'll do a little bit of review for Quiz 4. In the lab, we'll be discussing Lab 7, throat and urine cultures. I'll also talk about the way I'm changing the grading of the labs. Any questions about what we're doing? We're getting close to the end of the term. This is now week 5. At week 8, you'll be having the comprehensive final exam, July 23rd. And you'll be turning in your unknown project, July 21st. Your infectious disease project, you guys should be well along on it because that's due on July 14th. Any questions about any of that?
Is there a lab 8?
There is not a lab 8.
Okay.
Yeah, I decided to make you have the entire last week free for working on your unknowns project, and then you'll be working on your unknown project in week 6 as well. We'll start it, actually you'll start the unknowns project this week. All right, no questions about what we're doing. I'm going to close that down, get started on the lesson, move that over. So we're going to talk about nosocomial infections. These are also called healthcare acquired infections. These are infections the patient gets because they're going to a healthcare facility, like a hospital or a nursing home. And if they would not go to that healthcare facility, they would not come down with these infections. So these are something that are really bad because people are getting them when they're sick and they're coming to a healthcare facility. Any question about that?
Would that also be considered an opportunistic infection?
It can be. We're going to talk a little bit more about that, exactly what they are. They can be human pathogens that the patient picks up in the healthcare facility. They can be opportunistic infections. And that's really all of them. They're acquired because of the patient spending time in a hospital or healthcare facility. The patient did not have them before they came to the facility. 5 to 15% of all hospital patients, so about 10% on average, of hospital patients come down with a nosocomial infection each year. And we're talking about in the United States and in first world countries. It's actually higher if you go to third world countries. That's 2 million people a year in the United States coming down with a nosocomial infection. And that's per year. That's about 20,000 deaths per year in the United States from a nosocomial infection. It's actually the 8th leading cause of death in the USA, and that was before COVID-19. So it might be the 9th leading cause of death now, because COVID-19, I think I was just reading that that's now the #1 cause of death in the United States, COVID-19. But I'm not sure how they were looking at that. That might not be taking into account accidental deaths. I'm not sure. So don't quote me on that, but I just saw an article about that. Any questions about any of that? They are a real problem because about 1 in 10 hospital patients comes down with a nosocomial infection. And that's from the Center for Disease Control stating that. And if anything, they're conservative, meaning if it isn't a nosocomial, if they're not sure it's not a nosocomial infection, they won't count it as one of these. So that means about 10% of hospital patients come down with them. And then over 50% of nosocomial infections are preventable. That if we were taking steps to reduce them, we could reduce over 50% of them. It's a major economic burden on the United States. You don't have to know the numbers here, but it says $9.8 billion per year. And that's only for the 5 major infections. Nosocomial infections result from the interaction of 3 factors happening in the hospital setting or the healthcare facility setting. Meaning it doesn't have to be a hospital, it can be a clinic, it can be a, although people don't usually stay at a clinic, but a nursing home or what is that, a rehabilitation place. The first is that microorganisms are in the hospital environment. Hospitals are major reservoirs of human pathogens because when people become sick with a human pathogen, where do they go? To the hospital. So those diseases are found in the hospital. And there's no way you can avoid that. But some normal microbiota are opportunistic pathogens. And these are very difficult to prevent because everybody has normal microbiota and they can become an opportunistic pathogen. And then the worst part is some of the nosocomial pathogens that patients come down with simply by visiting the hospital or the healthcare facility, some of them are antibiotic resistant, so that when they're first treated with the first antibiotic, it doesn't work. And, you know, that might be if the patient is on the borderline of living or dying, and you give them an antibiotic to take care of the nosocomial infection. This may, and it doesn't work, that may tip them over. Any question about any of that? So microorganisms are in the hospital environment. It's impossible not to because normal microbiota will be with everyone. And then sick people who have human pathogens will be in the hospital. And then the patient is in the hospital for a reason. They are a compromised host, one whose resistance to an infection is impaired. It's impaired either by disease, by therapy in the hospital, or by burns or other procedures that have happened in the hospital or hospital-like setting, like a patient who has broken skin or broken mucous membranes and they come into the hospital. They're going to be deficient at being able to resist a nosocomial infection. So burns, injections, ventilators, urinary catheters, the administration of anesthesia, they all make the patient more susceptible to coming down with the disease, including a nosocomial infection. Some people have a suppressed immune system that will compromise the host. That can be from drugs the patient has taken, from radiation therapy that the patient is getting. Burns affect the immune system and immunocompromise the patient, as well as the fact that it breaks down the skin, so the patient's going to be more likely to get a skin infection. And then there's a chain of transmission of nosocomial infections in the hospital. The principal routes of transmission is direct contact, patient to patient. If one patient has a human pathogen and then they, are in the same room and they touch the other patient, that patient can come down with the human pathogen. There's also healthcare staff touching the the patient who's sick and then spreading the nosocomial infection to other patients, as well as visitors who can do that. And then visitors who bring into the hospital setting a nosocomial infection. And who do they most likely get? The person they're visiting. So it's probably their family members. This is especially true for special care units. Those patients are really immunocompromised, and they can come down with many nosocomial infections, including the normal microbiota that are opportunistic. It's very hard to stop those. And like I said, hospital staff to patients is a real bad one. If you are working in the hospital, you do not want to give your patients a nosocomial infection. There's also indirect contact transmission when the patient picks up a nosocomial infection by touching a fomite. Remember, a fomite is just an inanimate object, like somebody coughs or sneezes on their hand and then they touch a doorknob or a light switch, and then a patient in the hospital touches the same doorknob or light switch. They can get indirect contact transmission. Indirect contact transmission also occurs with urinary and IV catheters, contaminated surgical dressings, inappropriately handled specimens or tubes. Okay, the surgical dressing should never be contaminated. Use aseptic technique when you're in the hospital. You learned it in microbiology, put it into practice. And then there can be vehicle transmission, such as airborne transmission. In the hospital, if you have a highly contagious disease, it can spread by the ventilation system. Fortunately, that is not a major problem in hospitals. But when they have COVID-19 patients and they do cough, that can be picked up in the air, and then the circulation of the air in the room can be picked up by the ventilation system and then put into another patient's room, and that could be a problem. Any questions about any of that? So, microorganisms are in the hospital. The patient is there for a reason. They're a compromised host. And then there's a chain of transmission in the hospital or the healthcare facility. And that leads to nosocomial infections. Any question there? So how do we control nosocomial infections or healthcare-acquired infections? There are certain procedures that are generally implemented to control the spread of nosocomial infections. First, always use aseptic technique. You learned it in microbiology, now apply it when you're working in the hospital or hospital-like setting. You have to properly handle contaminated materials. Like if you're getting a specimen from a patient, you properly handle it so that the specimen is only in that container. You don't, get your hands contaminated, then touch the outside of the tube, and then let's say you wash your hands, but then you touch the outside of the tube. Now whatever else you touch will be containing that specimen. So use aseptic technique. Another way to control nosocomial infection is frequent and thorough hand washing. If you wash your hands frequently, you will reduce the spread of nosocomial infection. Something that should always be done is after you touch a patient, you wash your hands, especially before you go and touch a second patient. But you should wash them right afterwards so that you don't touch like a doorknob or a light switch and then spread it to a fomite and then somebody else picks it up. You need to educate the staff about nosocomial infections, and that is done now. You need to educate the patients and the visitors. And this is something that has only been done recently. Like I can remember, I don't know, 5, 10 years ago, when you went into the hospital, they didn't have signs up saying sterilize your hands before coming into the hospital, and they made sanitizer very readily accessible in the hospitals. And there's many other things you can do. You can read the online chapter notes, but generally work with your hospital or healthcare setting because they have a committee which is in charge of reducing nosocomial infections. And they're the ones who implement the procedures for controlling nosocomial infections, and then they monitor what's going on and how the hospital is doing. So, like I said, work with them on that. Any questions about that? Please learn nosocomial infection or hospital-acquired infections, because this is one of the most important topics you can learn from microbiology. Most of you are going to go work in the healthcare facility, and you need to know how to control nosocomial infections, because there's a lot of them happening in the United States. Any questions? We have controlled nosocomial infections about halfway from where they used to be, meaning we had about 50% more. And if you look at the old movies, when they show a doctor, when they listen to a patient's heart, they'd take their stethoscope, which would be around their neck, and they'd put it right up against the skin of the patient. And then when they were done, what did they do? They just dropped the stethoscope up against their chest, and they wore it proudly on their lab coat. That's what the doctors did. And they didn't realize that when they were listening to a patient's heart, they were picking up possible nosocomial infective agents on the stethoscope. And then medical profession themselves were spreading it because they never sterilized that stethoscope. And then when they would go to the next patient, they take the same stethoscope that had never been sterilized and put it up to the skin of the patients. And people have learned that simply by washing things, their hands, things like stethoscopes, after every patient's use, that they could reduce nosocomial infections by at least a half of what they used to be. The number one nosocomial infective agent used to be Staphylococcus aureus. Why? Because about 20% of Americans had Staphylococcus aureus on their skin, and it is a human pathogen. And so it was just being spread around in the healthcare facility because people weren't taking steps to control it. Nowadays, Staphylococcus aureus, because people are washing their hands and the stethoscopes and other instruments, it's not the number one nosocomial infection. But do you know what is? E. coli, something that everybody has, and yet it's the number one nosocomial infective agent in the United States. All right, if there's no further questions, let's move on to another topic. Epidemiology, the study of where and when disease occurs and how they are transmitted in populations. The CDC collects and analyzes U.S. epidemiological information. There are certain diseases that by law a healthcare facility or a doctor is supposed to report. So whenever a patient gets these diseases, they likely report it to the, the county health facilities, and then the county health facility sends it to the state health facility, Where is that in Arizona? Does anybody know where the state facility is? I'm guessing it's in Tucson, but it could be in Phoenix. Nobody knows? All right. And then the state facility sends it to Atlanta, Georgia, which is where the headquarters for the Center for Disease Control, the CDC, is. And they act as the repository for the United States. And then they publish that information, both the morbidity, the number of infectious cases, and the mortality, the deaths from the disease, in a weekly report. And it's called the Morbidity and Mortality Weekly Report, or the MMWR. And you can find that at www.cdc.gov. And they publish that each week what the notifiable diseases are. What's a little bit surprising is that the United States doesn't have one list of what diseases are notifiable. Each state is different, and by law, you have to report certain diseases to the healthcare facilities. So each state is a little different, but most diseases are in each state. I'm trying to think certain diseases that very few people get in the United States, like malaria. Not all 50 states require malaria to be reported, because the only way you get malaria, well, with certain exceptions, the only way you get malaria in the United States is when you go outside the United States and pick it up and then bring it back to the United States. Any questions about any of that? Malaria used to be spread in the United States along the Gulf states and the southern states and along the Atlantic. I'm not sure how far up it went, but it went at least as Washington, D.C. And how we got rid of that in the United States was they simply drained the swamps, reduced the number of mosquitoes. The mosquitoes that can spread malaria are found in the southern states and the Gulf states. And so every once in a while in the United States, mosquitoes will spread malaria. The last time it happened, I think it was in the 1960s, malaria came to New Orleans, and it was spreading in Louisiana. I'm not sure how far it went, but at least in New Orleans. Probably what happened was the patient brought back malaria to New Orleans, and then the mosquitoes in the region bit that patient and then started spreading it around. And I think how they controlled it was they just started spraying New Orleans with insecticides, and they reduced the mosquito numbers. And then they got rid of malaria once again in the United States. Any question about any of that? A little bit more about epidemiology. Morbidity, the incidence of a specific notifiable disease. The morbidity rate, the number of people affected in relation to the total population in a given time period. So you can look at the morbidity rate for Arizona for COVID-19, and they'll show that, the number of new cases. There's also the mortality rate, the deaths from notifiable disease such as COVID-19. The mortality rate, the number of deaths from a disease in relation to the population in a given time. And you can look at that for the incidence, or you can look at that for the total number over time. Any question about that? Any question about the principles of disease and epidemiology at all? If not, that's it for the lesson. So For those of you who want to stay, I will review for the quiz. For those of you who want to leave, you're more than welcome to do that. Any questions? All right. So let's review for quiz 4. It's going to cover chapters 13 and 14 and labs 5 and 6. So those are the labs we did last week and the lectures we finished today, but we mostly did last week. So let's begin with lab 5. Now I should warn you, I haven't made the quiz yet, but I'm in the process of making it. Somebody should remind me to get it up early so that students aren't surprised when I put it up and then they see it's due. It's actually in the schedule, and I don't know why people don't follow the schedule a little more. It's in the syllabus, the schedule, and so the quiz 4 will be due at 11:59 p.m. on Friday, whatever day of the week that is. That's going to be probably July something, but Friday of this week. So lab 5 was the cultivation of bacteria, know about the different culture medias, how to inoculate and how to isolate. Take a look at the learning objectives whenever you're going to have a quiz, because I'll take a look at these and we'll see if any of them are really important, and we'll give you a question if I think it's really important from the learning objectives. So understand the importance of proper specimen collection and transport to the laboratory. Explain the 5 principal steps used by microbiologists to process a specimen. And if I ask a question about that, I'm going to be very general because that's a fair amount of material to memorize. Differentiate between the various types of culture media. And so learn what media we've talked about and why they're used. Explain the purpose of a streak plate culture technique, i.e. streaking for colony isolation. So you should know that. And I remember there was a question about that in the lab, and a number of you got that question wrong. And we'll have to go take a look at our lab, because either something isn't quite clear. I did mention it. If you want to get a pure culture, you have to streak on 2 plates for colony isolation. First, streak for colony isolation, and then you take from an isolated colony, streak it out onto a second plate for colony isolation. And then when you pick through that second plate and get an isolated colony, when you grow that culture up, that will have a very high chance that it's a pure culture because it's gone through 2 colony isolation events. Any questions about that? And then define the following terms. You should know what a selective media is, a differential media, and to a less extent a reducing media. We haven't really used reducing media yet, so I'm not going to ask you about that, but I will about selective and differential media. And you should know that what a general purpose media is, as well as a selective and differential media. I mean, you can have a media that's selective, phenyl ethyl alcohol media is a selective media, 7% sodium chloride is selective, and then there's selective and differential. McConkey's is selective and differential. And then we've really only talked about one media, which is only differential, although you could call it a special general purpose media. You could also call it a fastidious media, and you can call it a differential media. And that's blood agar plates. Okay? That's the only one that we've talked about, which is only differential. At least the only one I can think of. Oh, wait, there is another one. There's a better example, too. And that's chocolate agar plates. That's only differential. No, wait a minute. I'm getting confused. That's not differential. It's general purpose. And fastidious, which I'm not separating fastidious media at this point. All right. So you should know how you isolate pathogens from a clinical specimen. And like I said, these steps: inoculation, isolation, incubation, inspection, identification, the 5 I's. I won't ask you a detailed question about that because this is a fair amount. And you can only read about it or virtually do it. But you should know what the general purpose medias are, and that is generally they're just used to grow up a lot of microorganisms. They're cheap. That's why we use them a lot in microbiology. They're not selective, so they grow many different kinds of organism. And we've talked about 3 different media. Nutrient agar, or Na, nutrient agar with yeast extract. It's a little richer, and that is also called Na. And then TSA, or tryptic soy agar. Those are all 3 general purpose medias. Now you can have an enriched general purpose media, and you use these for growing fastidious microorganisms. Fastidious microorganisms are ones that do not grow easily, and they will not grow on a general purpose media because they have specific nutritional requirements that have to be added to the media. And those special components are not in the media, they will not grow. And the only one we've talked about is, well, I guess we've talked about 2. Both of them have sheep's blood in them. One of them is called blood agar plates. You add 5% sheep's blood to the media, and the blood is added where the media is fairly cool, and so the blood stays in the media and the blood red cells are there, and you can use that plate to look for hemolysis, and that can be called CAP for, excuse me, not CAP, BAP for blood agar plates. And then the other enriched general purpose media is chocolate agar plates, or CAP. And it has blood in it, but the blood is added when the agar is quite hot, and so that lyses the red blood cells, and it makes the agar turn a chocolate color, and that's why they're called chocolate agar plates. They have no chocolate in them. It's actually blood that lyses. Any questions about enriched general purpose media? There's also selective media, and they contain a chemical or additive that inhibits the growth or halts the growth of some organisms. Two examples of that would be 7% sodium chloride added to the media, so a 7% sodium chloride plate. The 7% sodium chloride inhibits organisms that cannot grow in 7% sodium chloride. And then I mentioned phenyl ethyl alcohol. It has phenyl ethyl in it, and, well, phenyl ethyl alcohol actually in it, and it prevents the growth of gram-negative organisms, so they will not grow on a PEA, or phenyl ethyl alcohol plate. Now please understand, if the media is selective, the selection may not be 100%, and so other organisms may grow in it, but they generally will grow slower and less than if you were to compare it to a general purpose media growth. Any questions about that? So the selection is usually not 100% effective. There's also differential media, and they contain chemicals that allow you to see visible differences among different groups of organisms or bacteria that are grown on that media. The differences that are displaying are unrelated to how well the organism grows on the media. But by looking at the plate, you can see differences between two different species. There may be visible differences, such as the colony color or morphology, changes of the appearance on the medium itself. And realize, let me skip on to here, that there are media in more than one category. For example, McConkey's is both selective and differential. And we have a number of them that are both selective and differential. But McConkey's and EMB were the two that we'll run into most of the time that are both selective and differential. And then lastly, there's reducing media. These are media that you use to grow anaerobic bacteria, and they remove oxygen because anaerobes cannot grow, at least strict anaerobes cannot grow when there's low or no oxygen, and so you need to have something reducing in the media to pull the oxygen out of the media. All right, any question about that? And this is looking at McConkey's agar, selective and differential. Only gram-negatives grow on McConkey's, so it is selective against gram-positives. So if it grows on the McConkey's, it's gram-negative. And it also allows you to differentiate between those that are lactose fermenting. It has the sugar lactose in it, and a pH indicator. And if it ferments the sugar lactose, it'll make the growth acidic, and that'll change the color to a pink, and you'll see pink colonies. If there's a lot of acid produced, you'll see pink around the colonies, like right here, it's pink around the colonies. And the non-fermenting organisms will be off-white or whitish. These over here on the right. Blood agar is a differential plate and an enriched media, and you can look at this to look at the different hemolytic activity, beta hemolysis or complete hemolysis, where all of the red blood cells are lysed, and the white is where the bacteria is, the clear is where the complete hemolysis is. The dark ring is actually alpha hemolysis, and you usually see alpha hemolysis around beta hemolysis. Alpha hemolysis shown here, darkening of the red blood cells where there's partial hemolytic activity but not all of the blood cells lyse. That's why it looks dark or greenish on the agar. And then no hemolysis where the blood is good all the way in the media up until where the bacteria is. Gamma hemolysis is also called no hemolysis. Any question about that? So you should know.
Could you explain the difference between hemolysis and a cell that has been lysed?
A cell that has been lysed and hemolysis? If we're talking about a red blood cell, it's the same. Okay? Now, if you're talking about a bacteria, we don't say hemolytic activity of the bacteria, but a bacteria cell can be lysed. Like if you put it in soap, you'll lyse the bacteria cell. So does that explain it?
I think so.
Okay. So I think hemolysis is only used for red blood cells. And then lysis can happen to any type of a cell. So you should know the three general purpose media: tryptic soy agar, nutrient agar, and nutrient agar with yeast extract. And they're the general purpose media. You should know that if you add 5% sheep's blood to TSA, we call those plates blood agar plates, and that's a differential media and also an enriched media. Okay? Any question about that?
Is sheep's blood the only blood added?
No, you could get blood from like ox blood. I've never seen plates made of another species, but it would be possible to get blood from another species as well. Okay? Generally in the lab, you have to have it from an animal that you can get blood from, and sheep is one. And they call it ox, but the blood is most of the time coming from a cow, and I'm not sure. Oxen must be similar enough to cow that they use that term for both of them. And I'm not sure where the ox blood, if it came from cows or it came from an oxen, I don't know. But those are the only blood I've ever seen. I'm trying to think. You know, you certainly could get horse blood, but I've never seen it. And I've never seen human blood in a plate either, but there's human blood in hospitals. So like I said, it's possible, but I've never seen it. Chocolate agar is where you take the blood, you add it to hot media, and the blood cells lyse, and it allows you to grow some bacteria that require the ingredients from the blood, but these bacteria don't lyse the blood. Like the only one we'll really talk about in the lab was Haemophilus species, like Haemophilus influenzae. It can grow on chocolate agar because the blood is lysed, but it cannot grow on blood agar plates by itself. Sometimes what they do is they grow Haemophilus on a plate that has Staphylococcus aureus, and the Staphylococcus aureus lyses the blood, and that'll allow the Haemophilus to grow where the blood is lysed. But we won't. I'm not going to quiz you on that. Selective and differential media, you should know that McConkey's, eosine methylene blue, also called EMB, and we've only used Hectone agar, HE agar, once, but all of those are selective and differential, and they all are selective in that they allow gram negatives to grow, but not gram positive, and they're differential on they have lactose in them, and they'll be a different color if they ferment the sugar lactose. So if the growth grows, meaning if it's gram negative, you can look between those that ferment the sugar lactose and those that do not. Now, if it does not grow on a plate that's selective and differential, you cannot assay for the differential part. Is that clear? So if it's gram positive and it does not grow on McConkey's, you cannot assay for lactose fermentation. So it has to grow on the plate before you can assay for the differential ability of lactose fermentation. PEA is a selective media. It grows gram-positive bacteria. That's phenyl ethyl alcohol. And what happens is this alcohol gets in the gram-negative membrane and doesn't allow the outer membrane to form, and that prevents the gram-negatives from growing. Mannitol salt agar is another differential selective and differential media. It's selective for gram negatives, is that right? No, it's selective for organisms which tolerate salt. It has 7% bile salts in it, and that also is selective for gram negatives. They like to grow in bile salts because bile salts are in your intestines, and gram negative bacilli tend to grow in people's guts. Seems to me it's selective in one other area, but I'm not remembering. And it's differential in that it has the sugar mannitol on it, and it will be yellow if it can ferment the sugar mannitol. The pH indicator turns yellow if it ferments the sugar mannitol, and you ferment a sugar, you make acid byproducts. And that can allow us to distinguish between Staphylococcus aureus, which ferments the sugar mannitol, and other Staphylococcus species which will grow on mannitol salt agar, but will not ferment the sugar mannitol, and so they will not be yellow. All right, any question about any of those different media? You should know that Robert Koch developed an easy way to streak out a plate and get isolated colonies, thereby allowing you to get a pure culture. And you streak out the plate twice to get the pure culture. That was a great improvement over the method they had for getting out a pure culture before that. And it's such a good method for getting a pure culture that we still use it today. And what you do is, for getting streaking a plate, is you just get your bacteria sample, streak it out on a sector or quadrant, and usually you'll put down a large number of bacteria. You then sterilize your loop, put your second streak, that dilutes the bacteria. Do your third streak, you sterilize the loop again, and that reduces the number of bacteria. And then you sterilize your loop, and then you do your fourth streak. And in this case, we're now streaking isolated cells, and we have isolated colonies. Now, where the isolation happens does not matter. It can happen in the fourth quadrant, it can happen in the third quadrant, it can happen in the second quadrant, it can happen in the first quadrant. The idea is that you're just streaking and you're reducing the number of cells so that at some point you're streaking isolated cells so that isolated colonies will grow. Okay? It doesn't have to be the fourth quadrant. It can be any quadrant. Any questions about that? I've already talked about that. You should know the terms, and you should know what you did in the lab. All right, that's it for that lab. Let's move on. I don't think I'm going to finish this, but we can finish it tomorrow. Lab 6 was on disc diffusion, evaluation of disinfectants and antiseptics and antibiotic effectiveness. For this lab, we only did it for antibiotics. But one of the questions was, what else could you use this method for? And it tells you right in the title of the lab. You can use it on disinfectants and antiseptics. I mean, you can use it on other things, but that's a clear thing. So learn the primary objectives, identify the types of chemical agents and active ingredients used in various disinfectants and antiseptics, evaluate the effectiveness of disinfectants and antiseptics using the disc diffusion method, which, like I said, we never did that in this lab. We only did it for antibiotics. Determine the mode of action of various antibiotics and I'm not going to ask you a question about that one. Evaluate the effectiveness of various antibiotics using disc diffusion. I will ask you about that one. Okay? List the variables that influence the antibiotic disc diffusion susceptibility testing. Meaning, what do you do when you're doing the Kirby-Bayer antibiotic disc diffusion test method, also called the Kirby-Bayer test. And then you should know the terms sterilization, disinfection, disinfectant, antiseptic, bactericidal, bacteriostatic, antibiotic, spread plate, and zone of inhibition. So know the terms, they're defined right there. I'm not going to ask you about a table of the common disinfectants and antiseptics. So how you evaluate these, and we did it for antibiotics, is you put down on a plate a lawn of bacteria, and you watch the video on how they make those plates. And all you do is you streak the bacteria over the plate, and then rotate the plate, and rotate, I don't know if you can see this, you rotate the loop, and then you streak the plate again. And then you rotate the plate, like 90 degrees or 45 degrees, and you rotate your loop, and then you streak it on the plate again. The goal is to have bacteria go everywhere, so it'll be growing everywhere. And that's why we call it a lawn of bacteria. You have the bacteria growing as grass on a really good, healthy lawn, where the grass is everywhere. I mean, you don't want clumps of grass and then large patches of dirt in between. You want the lawn to be everywhere. Any questions about that? And how you evaluate the effectiveness is you then put the discs on the plate, and they have to be spaced out, and there are antibiotics in each disc. There are certain units, meaning each antibiotic has a certain number of units in the disc. And the table probably showed you how much it is, but I won't ask you about that. And then you measure the zone of inhibition around the disc where no bacteria were growing. And that's how you evaluate the antibiotic effectiveness. Now, when you measure that zone of inhibition, you have to compare it to a chart that tells you what size of the zone of inhibition it has to be for it to be sensitive to that antibiotic, and what size it has to be for it to be resistant. Okay, any questions about that? And that's how we measured the evaluation for antibiotic effectiveness. And by the way, if it wasn't resistant and it wasn't sensitive, then we called it intermediate. So antibiotic can be bactericidal if they kill the susceptible bacteria. They're bacteriostatic if they just inhibit the growth of the bacteria. And one of the questions in the lab was, how can you tell if it's bactericidal and if it's bacteriostatic? And hopefully you'll come up with the answer for that. It was in the lab, and if you didn't catch it, I talked about it when I was talking about this lab. So here we're looking at a plate with antibiotic disc on it. Let me see if I can blow that up just a little bit. And you can see a zone of inhibition. What you do is you measure that zone of inhibition. And generally it's best to take at least two measurements. Like this one, if you were to measure it there, it would be a little bit smaller than if you were to measure it here. And that's just because the zone of inhibition is cut off by the plate. This one doesn't look perfectly circular, so that one doesn't look perfectly circular either. So like I said, usually you want to take two diameters and then take the average. And that one, let's see if I can blow that up even further, looks like it has some colonies growing inside the zone of inhibition. So you still measure that zone of inhibition, but you make notes that one and this one have colonies growing. Any reason why there's colonies growing in here? Anyone have a reason? Why is that colony showing up? Anybody?
They're
Anyone
developing
have a
some
guess?
resistance.
Somebody's
There's
trying
some
to answer.
resistance.
You got to speak up.
Some resistance trying to be made.
Yeah, this cell is resistant to this antibiotic, but the majority of the cells are sensitive. And so we're getting the zone of inhibition, except for right there, where one cell grew and went on to form a colony. Okay? But that's exactly right. Let me shrink that down. So these are listing the different standardized variables that you have to have for the Kirby-Bauer procedure to enact. And there's a number of them. There's 6 on here, although some of them could be more than one. Like right there is actually more than one. But the point is there's a large number of variables that we try to control. And if you don't control them and you do the test twice, or 2 people do it in different places and they don't control the variables, they'll get different results. And we want to have the same results so that the test means something. And so we control the variables for when we're doing the Kirby-Bauer test. And I did mention last time that this is a typo there. The Mueller-Hinton-Agar plate should be 5 millimeters in depth. And if you have a different depth, the antibiotic will diffuse a different distance on the plate. And so everybody should have Mueller-Hinton media, and it should be exactly 5 millimeters in depth. And I don't know why this is wrong, and I can't go in and change it, so I'm just going to tell you. Whoever made it wrong. And then we're measuring the zone of inhibition there. All right, and then know how you did the lab. Any questions? So know how to read a caliper. So you read it from the 0 right here. So this one is 14 millimeters, not 9 millimeters. And then you, what is that? 9 millimeters of cephalosporin. And I think that's E. coli. And so we look on a chart, and 9 millimeters for cephalosporin mean that it's resistant because it's less than 14 or equal to 14. So this bacteria is resistant to the antibiotic cephalosporin when we look at this chart. Okay, any questions about any of that? All right, that's it for this lab. I have a few minutes left. Let's go ahead and talk about lecture 13. Obviously, you should know the terms of Chapter 13, understand the characteristics, structure, and classification of viruses, understand how viruses multiply, and they follow a life cycle. If they're a bacteriophage, meaning a bacteria virus, they can take the lytic life cycle or they can take the lysogenic life cycle to multiply. And remember, if you're following the lysogenic life cycle, you are a virus that can do either the lytic life cycle or the lysogenic life cycle, and you have to decide when you first infect a cell which life cycle to follow. Any questions about that? For animal viruses, they have the lytic life cycle, they have the budding life cycle, and a virus can go into a latent life cycle where the virus is not replicating. It's essentially metabolically inactive, and it's just staying there, and it can come out of the latent life cycle and then re-enter the budding life cycle and cause disease. And I guess there's also a persistent viral infection, which we never really talked about much. But that's another life cycle that animal viruses can have. Any question about any of that? And then know what prions are and viroids. So the goal of a virus is to replicate, and how they do that is they infect their host cell and then take over that host cell and make that host cell replicate for them. Okay, any questions about that? If they follow the lytic life cycle, they will lyse that cell, and that's how they release the viruses, the newly replicated viruses. So you should know the structure of viruses. They all have an internal molecule, which is their genome, and it's a nucleic acid. It can be either DNA or RNA. It's never both. And around the nucleic acid, there will be a protein coat that we call a capsomere. Some viruses have around the protein coat an envelope. We call them enveloped viruses. If they lack an envelope, we call them a naked virus. You should know the host range of a virus. That's the cells of a species that it can infect. Most viruses generally only infect some cells of one species. There are some exceptions, like the flu virus can infect, well, many different species. They can infect humans, they can infect pigs, they can infect horses. And what determines the host range is that the virus will have ligands that specifically bind to a receptor. It's usually a glycoprotein, but a receptor on a host cell. And so the virus has specific molecules for binding to its receptor. And that allows it to bind to a cell and determines its host range. All right, any questions about that? So I'm going to stop here, and at least I think we're going to stop a few minutes early. Yeah, 2:15. And then at 2:30 I will start up, and we'll talk about the lab for today. Any questions? If not, I'll see you in the lab. Did you want to talk to me about